Hello everyone and welcome to Pocket Size Bio of Dr. Casco. In today's video we're going to continue Chapter 8, an introduction to metabolism. This is part B of Chapter 8. So let's get to it. Let me just move to where we ended off. In the last video we ended off talking about how cells can do different types of work, right? And amongst this is mechanical work and transport work. So we're going to continue from there on to section 8.4. 8.4, enzymes speed up metabolic reactions by lowering energy barriers. So we're going to get into in more detail what enzymes are and how they work and the different types of enzymes too. So the law of thermodynamics, it states that what will happen and not how fast it will happen, right? So, we talked about spontaneous reactions and non-spontaneous reactions. So these laws of thermodynamics could give us an idea what type of reactions will forward easily and whatnot, but it doesn't tell us how fast that spontaneous reaction will occur, right? So this is something, that adds to a layer of complexity, with metabolic reactions. Exergonic reactions, they can be spontaneous. Of course, they are spontaneous, but they still can occur slowly. An example of this is sucrose breaking down into glucose and fructose, as you see here in this panel here. So sucrose, it hydrolyzes into these two compounds, glucose and fructose, and the free energy change would be -7 kilocalories per mole. But if you have this, if you have sucrose and you put it in a solution, right, if you put it in water, it can sit there for years, not dissociating at all, right? Not dissolving at all, breaking down at all. So what is used in biological systems are enzymes. And in this particular case, you have an enzyme called sucrase, and that sucrase can forward the reaction, can make the reaction proceed a lot faster in terms of seconds. Okay? So in general, that's the function of enzymes. And enzymes are biological catalysts, so they're chemical agents that speed up the reaction without itself being consumed in the reaction. So it doesn't become part of the product compounds, okay, or the product molecule. Enzymes, there's different kinds of enzymes. There's millions of protein enzymes. Okay, so the majority of enzymes are proteins, but you can have enzymes made up of RNA molecules that are called ribozymes, and incredibly important. They're crucially important for biological systems. And many reactions, without enzymes would occur way too slow for even the organism to survive. Okay. So related to enzymes is a concept called activation energy. And this activation energy is kind of a barrier for chemical reactions. So let's take a look at what that is. As you know, chemical reactions involve, molecules breaking bonds and forming bonds to create these new compounds. And when you want to break down sucrose, for example, you hydrolyze sucrose, and bonds are formed between the bonds between glucose and fructose. They must be broken down. New bonds are being formed with hydrogen and hydroxyl ions, so you can have these resulting molecules. And in order for that to occur, reactant molecules must absorb energy. Everything ultimately needs some amount of energy to break even bonds, right? Even if it's an exergonic reaction. So, my apologies. Reactant molecules must absorb energy from the surroundings. When new bonds of the products form, right, energy is released. You will have energy releasing for any molecule being created, bonds being formed. Okay. So one thing that's important to note is that for any chemical reaction, you see here, right? A and B and C and D, they need to react together. These particular molecules. You need a certain amount of energy for the reaction to proceed or to forward. Okay, that amount of energy that you require is referred to as the free energy of activation. Okay, or an activation energy, and is denoted by E sub A. Okay, it's the amount of energy necessary to push the reactants over an energy barrier so that the downhill part of the reaction can begin. And then you can start having new associations of bonds and between these compounds, these elements, these atoms, and form the products. Okay. Usually that energy that is provided is in the form of thermal energy. So the reactant molecules, they absorb that energy from the environment, from the surroundings. Because you're having this form of energy, there's more collisions, there's more force between the molecules, and that will agitate the atoms and within the molecules, enabling the bond breaking to occur, to happen. Okay. And when you're getting enough energy, when you're reaching that point of the energy of activation, which is shown here as that highest point in this little graph here of free energy, the molecules enter what is called a transition state. Okay. When you have enough energy that their bonds are about to break, the reactants are in an unstable condition, and that unstable condition is called a transition state. The bonds have enough energy that they're about to break, and you absorb enough energy that they actually break, and that's when you have this sudden change in state amongst the reactants driving into the products. Okay, so when the energy of activation is high enough, the transition, the transition state is reached, and then you have the products forming. And the transition state is rarely reached, okay, under standard conditions, standard temperature, and the reaction doesn't, in general, reactions don't proceed at all. Because imagine if everything would reach easily the energy of activation, chemical reactions will be occurring all the time, constantly around us. So you want to have a certain threshold, right, to keep in check chemical reactions. Okay. So reactions do occur generally when you apply enough heat. Okay. Some examples here: a spark plug will provide energy for gasoline and oxygen to react together and cause combustion. Okay. So without that spark, without that input of energy, hydrocarbons of gasoline, they'll be too stable to react with oxygen. Nothing will happen. Okay. So you need some initial form of energy to forward the reaction. Let's continue on here. Okay. So proteins, DNA, and other complex organic molecules, they're molecules that are rich in energy. And whenever you have reactions like hydrolysis coming in, this reaction is generally spontaneous, and it will release a large amount of energy for these particular products. But within chemical reactions, generally there's not enough energy at temperatures that are typical for the cell. So I'm talking about biological conditions, right? For a lot of organic molecules to reach this energy of activation. Okay, so take note of that. The majority of chemical reactions don't reach the energy of activation. And so it's quite interesting. The book kind of mentions, how are the barriers for these selected reactions surmounted to allow cells to carry out these processes of life. If we use heat, right, let's say we just increase our body temperature, it's going to speed up all of the chemical reactions. So you're kind of going to have the same situation where all of the reactions will occur, and you don't want that also. Another problem with this is that, as we talked about in proteins in the macromolecule chapter, heat can denature protein, and that's also a problem. It can destroy the function of the protein. It can kill the cell. So applying heat to everything isn't the best thing. So enzymes have, arose to speed up chemical reactions without necessarily any input of other forms of energy like heat, by lowering the energy of activation. And the interesting thing about enzymes is that they're very specific to to different compounds, as we're going to take a look at. So enzymes can help lower the activation energy to moderate temperatures. All right. I do want to note that enzymes do not change the free energy in itself, but it just hastens the reactions that will occur in any case. Okay. And again, enzymes are incredibly selective, very, very specific to a particular molecule. Okay. So they can determine chemical processes that will occur at particular times, depending on, concentration gradients and so forth, that we're going to take a look in the chapter, in chapter 7. Okay. In chapter 9 and 10, as we go along into cell respiration and photosynthesis. So I want to zoom into this particular panel here, the effect of an enzyme on activation energy. So in the previous diagram, I showed that the reactants need to, reach this certain energy of activation, or activation energy, either or is fine, to forward. When you apply in this solution the enzyme that would correspond to the reactants, you can see that the energy of activation greatly decreased, almost halfway there. Okay, so it's a lot lower, and this will enable the reactions to occur faster, just because the energy activation is lower. Okay, and then you will get that net output of delta G in an exothermic reaction in this particular thing. Okay, so that change in delta G is unaffected by the enzyme. Okay, so remember, you have the reactants here, and they have a certain amount of energy. So whenever you pass that threshold of the reactants, that liberation of energy is the same. So that's what I'm referring to as enzymes do not change that delta G. They just hasten the reactants to change into the products a lot easier. Okay, let's continue. All right. Talking about how enzymes work now. So substrate specificity of enzymes. Enzymes being, for the majority of cases, being proteins, right? Proteins, you guys know, they have their secondary, their polypeptide sequences, they have their secondary, tertiary, and quaternary structures. Enzymes, they have a particular site in which the substrate or any molecule that it reacts on, okay, the reactants will bind to. So you have any, generic substrate. Let's say in the case that we're using with sucrose, let's say this was sucrase, right? The sucrose molecule would come in and bind to a very particular place within the enzyme, and it'll look something like that. Okay. So I'm a little bit jumping ahead of myself. So whenever that reactant binds to the enzyme, it's referred to as an enzyme-substrate complex. Okay. And in terms of enzyme activity, you refer to those reactants that bind to the enzyme. They're called substrates. Okay. And whenever a substrate binds to an enzyme, is referred to as an enzyme-substrate complex. Okay. And as they're bound together, right, the enzyme and the substrate are bound, the catalytic action of the enzyme will convert that substrate to the product. The enzyme doesn't get affected, just the substrate. Okay. And you can see here kind of a situation that's occurring, this transitioning here. You have the enzyme plus the substrate, then you'll have that complex, and then you have the enzyme and the products. As we're going to talk about later on, these reactions are reversible. So that's why you see that there's a two-way kind of dynamics occurring. Okay, as the case of sucrase and sucrose, of course, hydrolyzing and so forth and so forth. Okay, so one thing that's very important to note is that enzymes are very, very specific to their substrate. And there's millions of substrates, so that means there's millions of enzymes also, right? The specificity of an enzyme, like I mentioned before, protein shape, comes from its dimension conformation, the 3D conformation. Okay, I won't go into so much detail with that, but of course, remember from the structures of proteins. The only usable region amongst the protein is that binding site. Now, these active sites, they can be called binding sites also. They can be called pockets or grooves. So make sure you know these terminologies also, right? And they're generally on the surface of a protein. In this particular location, you have this active site where you have the substrate coming in this example. Okay, so here the book gives this example of a hexokinase, right, binding to its substrate, which is glucose. Okay, but understand that idea: active site, substrate, and enzyme. Interestingly, the active site is usually formed by a few amino acids. So the majority of the protein molecule and these long sequences that we've seen, right, in polypeptide formation, will create these alpha helices and create these, this beta pleated sheets and these 3D structure. In the case of enzymes, to ultimately create this active site. You're going to use all this chunk of protein to create that framework to configure and have a very specific active site. It's quite interesting there. Okay, the specificity of an enzyme is due to the fit between the active site and the substrate. All right, we're gonna look at this in a little bit more detail, but the active site itself is not a rigid structure, right? It's not like a solid cement structure that you're just gonna plot in like a Lego, right? So it is breathable, if I can say, and it is a little bit flexible. Okay, we're gonna see that induced fit later on. So the interactions between the chemical groups on the substrate and those of the amino acids, the side chains of the amino acids, will ultimately cause a slight change in the enzyme shape. Okay, this is called induced fit, where the enzyme can actually, come in and cover and tighten the grip on the particular substrate. Okay, and then that will enable the bonds to become a little bit weaker. There's more tension growing on the bonds, and then they ultimately break. We're going to see this in a little bit. Okay, so the catalytic activity of the active site we're going to jump into now. Okay, so substrates are generally held in the active site by weak interactions. You guys know weak interactions: hydrogen bonds, and ionic bonds, and ionics not so much, but hydrogen bonds. And the R groups of those amino acids on the active site will help in that catalysis of substrate to product. Okay, so a single enzyme can catalyze thousands of substrates, so thousands of reactions a second. Okay, again, as I mentioned before, enzymes are unaffected by the reactions, meaning that they're reusable. It can be used many, many, many times. Okay. Sorry, I've been having a terrible cough and throat pain. Okay. So most metabolic enzymes can catalyze a reaction. Catalyze a reaction in both the forward and reverse direction. So if you remember in the previous little diagram, schematic of sucrase going into the complex and then the products. This is what I'm referring to here. So the direction on the depends on the relative concentrations. Okay, so enzymes, they catalyze reactions in the direction of equilibrium. So where there's a higher concentration and you want to promote equilibrium, they're going to gear toward that direction there. Okay. Of course, remember we talked about stability and the idea that the cell doesn't want to reach equilibrium. Of course, the products then will be going on to another set of reactions. Okay. So in reactions involving more than one reactant, the active site brings substrates together in the correct orientation, and then the reaction can proceed. Now, before I get into the diagram, I just want to kind of note this. So what the enzyme actually does, when the substrate binds to it, it warps, it kind of alters and stresses the bonds of the substrate, right? So it can get weaker as it reaches this transition state. So there's a lot of warping, right? So there's a lot of stressing and bending of critical bonds that need to be broken in order for the reaction to forward. So those R groups that are involved in that active site will create a little microenvironment to help tension the substrate. Okay, and this is essentially what I'm talking about here. Okay, you have these two substrates. You can have more than one substrate working together. They come into that active site. The enzyme, initially, looking like this, can come in and it alters its shape a little. That induced fit situation. Okay, so substrates enter the active site. Enzyme changes its shape to its active in such a way that the active site will kind of engulf a little bit the substrate. Okay, held down by weak interactions, this will activate this will weaken and lower the activation energy. Because of that, the reaction will be able to proceed, and those initial substrates will turn into the products there. Products are released, enzyme returns to its initial conformation. Okay. And again, what enables this is R groups. Okay. Now, sensitivity of enzymes is very important to the environment. Okay. We're going to see that in a little bit. But active sites, again, they can have their little microenvironments. You can have areas of low pH, and that low pH can enable hydrogen ions to transfer from to the substrates. Okay, which will help hydrolysis or changing, just catalyzing the reaction. Okay, some enzymes can briefly bind covalently. This might be a little bit of overkill of information, but just to know, right? They can bind covalently to substrates, and later on you can restore those side chains of the amino acids to go back to their original conformation. The rate at which the number of enzymes can convert, the substrates to the products will depend on concentrations. Okay. Of course, if you have a low substrate concentration, you can speed up the reaction by increasing the substrate concentration, right? That can be used into the available binding sites. If you have a fixed amount of enzyme quantity. Okay, if you have a surplus of substrate concentration, you're going to reach a maximum that you cannot speed up the reaction anymore if you have a fixed number of enzyme molecules, just because all the enzymes are being used at a certain amount of time. Okay, so you're going to reach a threshold. Okay. So all the enzymes are engaged, right? So the enzyme is saturated. You reach a kind of saturation point. Okay. So the rate of the reaction is determined by the speed at which the active site can convert substrate to the product. Okay. In that particular case, then you just need to add more enzyme to speed up the reaction. Okay. This is a nice little scientific skills exercise just to take a look at how enzymes work, concentrations of inorganic phosphate, rate of ATP, and a certain amount of time. Okay, so you can pause the video there so you can see this scientific skills exercise. Okay. Effects of local conditions. So I want to highlight this. This is I've seen this on tests. Very standard and critical thing to note. Basically, how the environment affects enzyme activity. Okay, definitely, enzyme activity is very sensitive to the environment, to environmental conditions. What I mean by this is temperature and pH mainly. Okay. So different enzymes have different tolerance ranges and optimal ranges in which it works best. And at these conditions, you'll have the most active conformation and highest rate of efficiency. Okay, talking about temperature, of course, as temperature increases, so this little shortened form INC increases, there's more collisions, right? There's more collisions between substrates and active sites, so the reaction will just, plainly just go faster, just because there's more interactions. The further increase in temperature, though, thermal energy begins to disrupt the weak bonds that will stabilize the protein itself. What happens? The protein denatures. Okay, so if you remember that from just general principles of protein structure, temperature is an important player in enzyme activity, denaturation. Again, as I mentioned before, each enzyme has an optimal temperature that will allow greatest number of collisions, fastest conversion from reactants to products. Let me just go down a little bit here. Most human enzymes have optimal temperatures between 35 to 40 degrees Celsius in our body. Of course, this is our normal body temperature, and this is why it's so important to lower down to lower fever, because you're going beyond the threshold in which enzymes can work effectively. Okay, different organisms have different enzymes that are specialized at different temperatures. You have the thermophilic bacteria that live in hot springs. They have enzymes that are optimized to work at 70 degrees Celsius or higher. Okay, so that's what I'm referring to, and this is very specific to enzymes. Just to give you a sense here, right? And again, enzymes do have like an optimum range, right? It's not just a fixed number. You do have a tolerance range where you have optimized work here. Human enzymes around 30 degrees Celsius, right? Then as you get out of this range, it quickly goes down, the rate of the reaction. Heat-loving thermophilic bacteria, 75 degrees. As you get out of that, the reaction rates will go down. Okay. Let me just go here. Okay. Talking about pH, right? Active conformation of an enzyme requires a particular pH. So generally, for many enzymes, you have pH ranges going from 6 to 8. Okay, for most enzymes, but we do have some specialized enzymes, for example, in the stomach, that work best at pH ranges of 2 or pH levels of 2, or the intestines that have an optimal pH of 8. Right? If you get out of these, the efficacy of the enzyme will decrease. Okay, so an example of that stomach enzyme, you have pepsin. Okay, you have trypsin for intestinal enzymes that you can see their optimal efficacy is around their particular pH ranges. Okay. Environment. So in terms of temperature and pH, there are other things that are occurring within the environment that can help enzymes work effectively. There are non-protein helpers, little chemicals that are called cofactors or coenzymes. I'm going to highlight that. So cofactors, they can bind permanently or temporarily to an enzyme. They're usually inorganic cofactors like zinc and iron, copper and or copper in the ionic form that can enable or they can activate the enzyme to work, right, to bind to the substrates. Cofactors. You can also have organic cofactors. They're referred to as coenzymes, and many vitamins belong to this category. Are coenzymes, and they help bind to the particular molecule or the particular enzyme, again, to have its catalytic activity in the active state. So it's important to, have your vitamins and your minerals, again, for efficacy of enzyme work. Okay. Opposite to working, sorry about that, opposite to increasing the work of the enzyme, you have molecules that can inhibit the functionality of the enzyme. Okay, they're referred to as enzyme inhibitors, and these are very selective, very specific molecules or chemicals that can stop the action of enzymes. And if the inhibitor, so there's certain conditions here, if the inhibitor covalently bonds to the enzyme, then the inhibition may be irreversible. So you might need to, you won't be able to use it anymore. If the inhibitor performs weak bonds, right, then the inhibition may be reversible. Okay, and some reversible inhibitors, they resemble the substrate. They look almost identical to the substrate. And because they look almost identical to the substrate, they can compete for the active site. These inhibitors are called competitive inhibitors, okay? And depending on your substrate and inhibitor competition is all a matter of concentration, right? Within the particular solution in the cell. Okay, as you can see here, hey, you have normal binding. This is a normal case. You have an enzyme in your substrate binding to the active site. Competition, you can have a competitive inhibitor where it doesn't have to have the complete, pocket shape. But enough that it can bind to a particular area, that's already competitive inhibition. There's another form of inhibition that is called non-competitive inhibition, where these inhibitors, they come in and they can bind to another part of the enzyme. And whenever they bind to another part of the enzyme, this will cause the enzyme to change shape. And if it changes shape, it can alter the active site of the where the substrate is supposed to be. So because it changes the active site shape, it cannot bind to the substrate anymore. So this is non-competitive inhibition. Okay. Of course, the enzyme becomes less effective. Some examples of these are toxins and poisons, okay, which are often irreversible enzyme inhibitors. Okay, sarin, a nerve gas that was released by terrorists, unfortunately, in Tokyo subway in 1995. They released this gas on a train, which did this type of reaction there, right? There was a covalent bonding occurring between these toxins to the R group of an amino acid, serine, that decreased efficiency of the enzyme that it was, targeting. More particularly, acetylcholinesterase. Okay, it is an enzyme that focuses signaling. Sorry about that. Okay, acetylcholine. Okay, neurotransmission. All right. Another example is DDT. So it acts as a pesticide by inhibiting key enzymes in the nervous system of insects. So you have another situation here. So there are many examples, right, for insects, humans, pesticides, antibiotics works in a similar way. Inhibitors, they inhibit specific enzymes for bacteria that can alter the function for synthesis of bacterial cell walls. Okay, penicillin. So while they can have permanent damage, not all of them, again, are metabolic poisons. So we use them; they're useful in our body. Okay, depending on what you want to inhibit. Okay. So molecules are naturally present in the cell, and they often regulate enzyme activity by acting as inhibitors. Okay. Let me go to the next page there. Okay. So here you have evolution of enzymes. So biochemists have discovered and named more than 4,000 different types of enzymes of different species within different species. Okay, and there's of course many more. And this diversity, we're gonna see this in a lot more detail when we get into mutations, right? Enzymes, as you know, are proteins that are made from polypeptide sequence structures, which are ultimately coming from amino acid sequences, which come from genes, right? And if you have a permanent change in a gene, we refer to this as a mutation. So you're altering the amino acid sequence just because you're altering the DNA sequence within a particular gene. So it can happen that mutations, they change the particular amino acid sequence of enzymes, sometimes altering the active sites, okay, or key domains or regions that enable the conformation of the protein to come about. Okay, but at the same time, it doesn't necessarily have to be a bad thing, where you can have novel activity, and you can have a new type of active site being created, or it could bind to a different substrate. So it doesn't necessarily have to be a bad thing. Okay, so under environmental conditions, a new function can arise, so that could benefit the organism. Natural selection would then favor that mutated form. It might be more efficient, so it could persist, it can last within the population, and ultimately, generations and generations of a particular population will now contain that altered form. Okay. Researchers have used a lab procedure that mimics evolution under natural conditions, populations, for enzymes to see how this actually works. Okay. There have been many lab experiments to simulate that idea of evolution through repeated mutation and selection for bacteria, right? In E. coli, there's an enzyme called beta-galactosidase, which was experimentally evolved to act as a new disaccharide. Okay, it was that's very interesting there. Beta-galactosidase breaks down the disaccharide lactose into simple sugars like glucose and galactose. So researchers would introduce a mutation on this E. coli gene and test if the bacteria would be able to, break these same things or some other types of disaccharides. Okay, so they tried it, repeated it many times, and after these kind of mutations, that they would add mutations, they saw that this evolved enzyme would be able to bind to several to substrates, to other substrates, several hundred times more strongly, okay, than the original enzyme. So it's quite interesting how they kind of experimentally provided a way to see evolution. Okay, let's continue on. All right, almost finishing off. This is the last section of chapter 8. So 8.5, regulation of enzyme activity helps control metabolism. So metabolic control often depends on allosteric regulation. So we're going to take a look at that. Many molecules that are naturally regulate enzyme activity, they behave like reversible non-competitive inhibitors. Okay, so you have the situation of allosteric regulation. So in this particular case, let me zoom in here. Sorry about that. A protein's function at one site is affected by the binding of a regulatory molecule. I'm going to highlight this because this is important. Okay, a regulatory molecule to a separate site. Okay, to a separate site. So inhibition, this can inhibit or stimulate an enzyme activity. So it's called allosteric regulation. Okay, and these allosterically regulated enzymes, they're composed of 2 or more polypeptide chains. As you see in this particular example here, you can have these little substructures of the enzyme. Okay, let me just move down a little bit. So the complex of having these regulatory molecules can oscillate between being a catalytically active form and an inactive catalytic form, and an inactive form here. Okay, so an activated or inhibiting regulatory molecule can bind to that regulatory site. This regulatory site, it's usually called the allosteric site. So that's a more technical term to note. Okay, your allosteric site. And these sites often are located where subunits are being joined in there. Okay, so just to kind of show you here, you have an enzyme that is in active form. Okay, you have its active site, and you can have multiple active sites. Then you can have your regulatory site, your allosteric site. Okay, and then you can have your inactive form. Okay, where you have the non-functional active site, so it has a different shape there. Okay, and so forth. So it can bounce between these two states. So whenever you have these this active form, and you want it to stay in this active form, you would have these activators or inhibitors that we're going to take a look at now. So you have an activator. So an activator stabilizes the conformation to be in the active form. So you have this little molecule here. It binds to those regulatory sites. And now the substrate is kind of in an induced state that it can collect the substrates and coming in, and you can have multiple of the same substrates working for more efficiency of reactions to occur. Or you can have, in the inactive form, you can have an inhibitor that will stabilize the inactive form of the enzyme. So if you notice here, you have this situation here. Okay, the inhibitor will come in and bind to that regulatory site, and so it's permanently in this kind of deformed state, this inactive state. So you cannot get the substrates to come in and bind at all. Okay. So the subunits of an allosteric enzyme, they fit together in such a way that the shape will change, as I mentioned before. Okay, and the change of one subunit can actually affect all other subunits. So if one changes, everything is going to change. A single activator or inhibitor molecule that binds to one regulatory site, so remember that, okay, will affect the active site of all the other subunits. Okay, now this is regulated depending on the chemical conditions of the cell. If the cell requires the enzyme to become more in the active form, then you can have other enzymes that will come in and that can displace this this activator or this inhibitor, depending on the cellular conditions. Okay. So by binding to key enzymes, reactants, and products, of ATP hydrolysis, they actually play a role in balancing the flow between these enzymes and, of course, ultimately in metabolic pathways. Okay, so in ATP in itself can bind to several catabolic enzymes allosterically, so they can act as inhibitors in their activity. Okay, and ADP can function as an activator for the same enzyme. So this is what I'm referring to as activator and inhibitor, that depending on the cellular conditions. Okay, for catabolic pathways, but they could be used for anabolic pathways also. Okay, so allosteric enzymes, they can control the rates of key chemical reactions, right? Sorry about that. You do have another particular situation called cooperativity. And in this case, so you have enzymes that have, with multiple catalytic subunits, and that they're bonded by a substrate molecule to one active site, and a multiple subunit enzyme can trigger the shape change in all subunits. Okay? So this is the idea of cooperativity. So you have an amplification in the response of the enzyme to the substrates. Okay? You have an amplification, so a more efficacy for the enzyme to work. Okay, this will enable more enzymes, the enzyme to collect additional substrates. Okay, so this is just the substrate itself. So you have an enzyme, and in inactive form, a substrate can just bind to one of the inactive forms, which will create a cascade to change the other kind of locations for the substrate, the other active sites. Okay, there's another type of allosteric activation. Nothing to do with inhibitors and activators, just the substrate itself. Okay. The vertebrate oxygen transport protein hemoglobin would be an example of cooperativity. Okay. Hemoglobin is made up of 4 subunits, each with oxygen binding sites. So this would be an example of cooperativity. Okay. This might be a little bit too detailed, but I'll just come in and mention here. Yeah, so again, the concept of hemoglobin as cooperativity, it's made up of 4 subunits, as I mentioned before. Oxygen will then bind, come in and bind to these binding sites of hemoglobin, and so forth. So you'll be getting more oxygen able to be transported. Here, under conditions of low oxygen, as in oxygen-deprived tissues, hemoglobin is less likely to bind to oxygen. Okay, so then it releases it whenever it's needed. Okay. And you'll allocate to the tissues that are more vital. Okay. When oxygen is in higher levels, as in the lungs or gills, the protein has greater affinity to oxygen, so more binding sites are filled. Okay. More use, more efficiency, more work for overall collection of oxygen to be transported to other tissues. So allosteric regulation, right, is likely widespread but difficult to study due to low affinity binding. So now this goes a little bit beyond the scope of Campbell biology, but there are rates and certain conditions for low and high affinity binding for allosteric regulation. Okay. Allosteric regulatory molecules, they tend to bind the enzyme at low affinity and are thus hard to isolate for research, right, or experimental studies. And this is interesting for a hotspot for studies in pharmaceutical science, right? They're turning their attention to allosteric regulators, which exhibit higher specificity for particular enzymes than do inhibitors that bind to the actual active site. Okay. Okay, last part here is a common method for metabolic control. You see this a lot. We're going to see this a lot later down in negative feedback and homeostasis. Feedback inhibition. Okay, common method of metabolic control is feedback inhibition. So an early step in metabolic pathways, right, is switched off by inhibitory binding of the pathway's final product, okay, to an enzyme acting early in the pathway. Okay. What I mean by this, so you have your initial substrate. This initial substrate will go through a metabolic pathway and it will undergo several changes, okay, with different enzymes. Threonine you have in this active site here. As it'll change, it'll go to an enzyme 2, intermediate, structure, and so forth and so forth and so forth until you get an end product. In this case, isoleucine. Sorry about that shaking. As you get enough of this isoleucine, enough concentration, this isoleucine can actually work to stop the reaction from proceeding. Okay? This is called feedback inhibition. The final product of a metabolic pathway can come in and be used as an inhibitor for that same enzyme if you have enough of that final product, right? This is referred to as feedback inhibition. And it prevents a cell from wasting chemical resources by synthesizing more product than is needed. Whenever you have enough of that product and you carried out a subsequent reaction, this product can be used to stop its own production. So it's quite interesting in that sense. Okay, and this does involve, right, the localization of enzymes within a cell helps order metabolic reactions there. And this is kind of going back to the cell with compartmentalization, right? The organization of cellular structures help bring order to metabolic pathways. And if you have a set of enzymes, that consist of several steps, they can come in and you can have a multi-enzyme complex, right? So you can have the product of one and then moving to another one and then moving to another one. And the product of the first reaction becomes the substrate of the adjacent enzyme, as we see, as I kind of went a little bit ahead here, as you see here, right? Until the final product is released. And then the some enzymes and enzyme complexes, they tend to be fixed within the cell. Very clear or key example there is like cell respiration on the cell membrane, right? You have many commenplexes there. Or they're often confined within membrane enclosed organelles, right? Within the lumen or the fluid inside other organelles. Okay, so you have a lot of kind of synchrony amongst metabolic reactions, even amongst different organelles communicating with each other. So metabolism, right, is an intersecting set of chemical pathways, a very critical characteristic of life, right? They even mention a choreographed interplay of thousands of different kinds of cellular molecules. Okay. As I mentioned before, right, cell respiration. Very a nice example, crucial example that we're going to get into detail in the next chapters on how you can see this with specific molecules, right? So with that being said, this finishes chapter 8. Okay, take a look at the chapter review there to get a sense of what we talked about. Okay. Testing your understanding. I will start to make Quizlet flashcards on video, so hopefully that could be helpful for you also. And post them on YouTube. I might have moved it a little bit too fast there for you guys to see. Okay. All right. The next video, we're going to go over chapter 9, which is cell respiration. and fermentation. Okay, that's it for the video. I hope you found it useful. I do apologize for some of the stuttering that I was doing throughout the video there. And thank you for watching, and I'll see you in the next one. Bye-bye.